Before you can do any kind of research, you're going to have to create your own methods for conducting that research. So there's a few important things to consider when it comes to your research method. First of all, sampling. When I'm talking about sampling, I'm talking about a small group of individuals that are taken from a larger group. We call that large group the population. So a population would be every member of some kind of thing. And in psychology, the population we're interested in is humans. So we draw a small group of humans to then try to make, predictions about human behavior in general. And when you draw a sample, it might seem appealing to get what's called a random sample. That just means every member of the population has an equal chance of participating in this experiment. So while that might sound appealing because, it's random, everybody gets chances, the world is a big place. If you wanted to truly draw a random sample, that would mean you would have to give every single person living on the planet right now an equal chance of participating in your study. And if your study's in Los Angeles, that's going to require some significant travel for some people. So generally we don't do random samples in any kind of science. What we try to do instead is draw a representative sample. So we look at the population as a whole, try to get a sense of what kinds of individuals are in that population, and then get one of each kind. So if you have, like, let's say you want to do research on a group of Caucasians, right? Your sample should involve Caucasians. But if you want your conclusions to be more broad, to apply to all kinds of people, then your sample should have representatives of all the different kinds of people that you're interested in. So it's just, all I'm saying is a representative sample has individuals from all different types that exist in the population. That's what we tend to try to do. Unfortunately, quite often sometimes we will get what's called a biased sample. So that just means the sample we've drawn either excludes certain kinds of individuals, or it just represents one of the kinds too much. Like we're getting too many of this one kind of person and we need more of this other kind, basically. So sometimes, unfortunately, biased samples can, skew the data. Another important consideration when developing your research methods is variables. And there's two really important kinds of variables for you to remember. You have the independent variable, or the IV. This variable, another word for it is factor, is the variable that is manipulated by the experimenter. So the experimenter decides if they're going to give you one version of this factor or a different version of that factor. The second kind of variable would be the dependent variable, and this is a factor that's influenced by the independent. So all that means is, depending on which version of the independent variable you got, the researcher is going to be interested in how that affects you. So if they gave you this one, they want to see the effects of that. If they gave you the other one, they want to see if there's different effects. So that's how independent variable and dependent variable work. Independent potentially has an effect on the dependent. Now, unfortunately, this is made a bit more complicated by the presence of extraneous variables. There's always going to be extraneous variables, because extraneous variables I'm just talking about things that can affect the dependent variable, but they're not manipulated or controlled for by the experimenter. So these are, like, you could call them confounds, you could call them problems, you could call them whatever you want, but they definitely make the researcher's life more difficult. So here's just a few examples in this table right here. You can see I have a list of independent variables on the left and potential dependent variables on the right. So for example, depending on which drug I give you, or depending on which kind of therapy I give you, we would want to see how it affects you. Like, what are those treatment outcomes? Another one would be diet. Like maybe I want to do a study comparing 3 different diets. So I assign one group to diet 1, a different group to diet 2, and another group to diet 3. And then after a couple of weeks, I would just check their weight and see how much weight they've lost. Maybe I'll find that one of these diets is just better at, getting people to lose weight than the others. Now, a special kind of variable is called a quasi-independent variable. You could also call it a demographic variable, because these quasi-independent variables, they separate people into groups just like a real independent variable would, but you can't manipulate it. These would include things like a person's age or gender or ethnicity or sexual orientation or religion, things like that. So it's things that definitely put the participant into a separate group, but it's not like you as the researcher get to decide. It's like if I want to do a study on gender differences, those groups have already been made. It's not like when you come into my lab, I'll be able to say, okay, for the purposes of this experiment, you're going to be a female and you're going to be a male. And that doesn't make any sense. That's why it's quasi-independent. So you can't really do true experiment using quasi-independent variables, but you can still come to some good conclusions in general. So when I'm talking about true experiments, I'm talking about generally laboratory experiments, and that's just one of the different kinds of research methods that are out there. So here is an illustration. Here's a chart. So the 2 basic kinds of research you can do are either experimental or non-experimental. So with experimental research, you have that true research. You could also call it laboratory research. And then there's quasi, independent research, research using those quasi-independent variables. And then there's field research. Whereas with non-experimental research, you have things like surveys, observational studies, meta-analysis, archival analysis. There's many of them, but the key idea of these non-experimental studies is there are no independent variables. So you can't really see how they affect the dependent. So with true experiments, you have an independent variable, you have that manipulation, and you are observing its effects on the dependent variable in a controlled setting. But remember, with a quasi-experimental study, with these quasi-independent variables, you can't manipulate the independent variable anymore, but you can still observe its effects in a controlled setting. But with a field experiment, you can still manipulate the independent variable and observe its effects on the dependent, but you're going to have to deal with a whole lot of extraneous variables due to the fact that you are not in this kind of controlled laboratory setting anymore. You're out in the real world where all kinds of things can happen that you would never be able to predict. So the reason why you would want to do any kind of experiment, though, is to see, does this independent variable, does this, factor have an effect on my dependent variable? So you're looking for cause-effect relationships. So if you ever want to know, what would X do to Y, well, you're going to have to do an experimental study on that. So with an experimental study, the basic idea is you're going to have at least 2 groups. Typically one's like a control group that isn't really what you're interested in, and the other one would be the experimental group that's getting that, version of the independent variable you are interested in. And you just treat them both the same way and see if there's any differences. Like maybe you give one group of participants this new drug you've been developing, and you give another group some other drug that's already on the market. And by comparing how they affect the individuals, you'll be able to see if your new drug is better or as good as the one that's already out there. So it's all about just comparing this experimental group to your control group to see if there's any differences. Now, you want to make sure that these 2 groups are as similar as possible and are as treated as similar as possible to try to eliminate those extraneous variables. And one of the easiest ways to do this is through random assignment. So that just means when the participant walks into the lab, they are randomly assigned to either that experimental group or the control group. So just simply doing that will be a great way to reduce extraneous variables. Non-experimental research, on the other hand, is about identifying correlations. And a correlation is just a relationship between two variables. Like if I tell you that two things are correlated, that just means they change together. You know, as one variable increases, the other variable increases. Or maybe one goes up and the other one goes down, you know. But the whole idea here is they're somehow linked, it seems. But it's really important to remember that just because they appear to be linked, that doesn't mean that one causes the other one to change. That might be the case, or it might not. Sometimes it's impossible to tell. So you should never try to determine that kind of causation link from doing a non-experimental study, because the data just simply cannot show that. All the data is going to show you is what's called a correlation coefficient. That's just a simple number from +1 to -1 that describes the relationship. So the closer that number is to a +1, the stronger the relationship is, and the more positive it is. So a strong positive relationship would mean that every single little change in one variable will have similar changes in the other variable. So if one goes up a little bit, the other one is also going to go up a little bit. The closer that correlation coefficient is to -1, the more strong it will be in the negative direction. So when one variable goes up a little bit, the other variable would go down a little bit, for example. But if that number is close to 0, whether it's positive or negative, that just basically means there's no real relationship here. You know, one variable might change dramatically and the other one is not really going to do much. So the correlations we're interested in are the ones that are either really strongly positive or really strongly negative. But remember, correlation is not causation. Correlation just means that these two things appear to be linked. Just because they're linked, that doesn't mean that one causes changes in the other. Like, for example, if we find that variable A and variable B have a correlation, it might be the case that A influences B. Like, changes in A will affect B. It could also be the case that changes in B affect A. Or maybe there's a third variable that we don't even know about, a variable C, that affects both A and B. So changing C would affect both A and B. Like, there's so many different possible solutions to this puzzle. It's impossible to know unless you do, you know, true experimental research. But one of the most talked about correlational, you know, studies that was done, where people have made this kind of causative inference, is of course MMR vaccines and autism. I'm sure you've heard about this potential link. And let me just say a few basic things. I have a couple links to videos in the description below if you're interested to learn more about this. But just to give you a little bit of background of what actually happened, there was a study that looked into a potential correlation between children developing autism and receiving MMR vaccines. And that study did publish evidence of a correlation. But unfortunately, people latched onto this, you know, the fear set in, and they just assumed that because these things appeared to be related, then that must mean that the MMR vaccines have caused the autism. And that idea, that fearful idea, was propagated and propagated thanks to perhaps well-meaning but ignorant celebrity figures. And the end result is that now people, because they've heard it so often, people think that this has some validity. But remember, I said it was one correlation published in one study, and to make things even worse, they later found out that that study lied about it. So that study had falsified their data. There is no link. Not only is it not a causative link, but it's not a link at all. So all of this is just a bunch of BS, to put it simply. It's paranoia. It's fear of something that just simply does not exist. Getting back on track. When you're doing a non-experimental study, there's different kinds you could do. Like you could do a survey. I'm sure you've experienced a lot of surveys yourself. You know, generally it's pen and paper, or nowadays it's on the computer, like you saw at the beginning of this video, where you're just being asked a series of questions and responding, and usually like a multiple choice format. But you could also do what's called an observational study. With an observational study, it's exactly what it sounds like. You're watching people and taking notes. You could also call this a naturalistic observation. So these are the people watchers. You know, they just have like a notebook and they're recording like how often do people do whatever it is they're interested in. A meta-analysis, that's when you are interested in developing a theory. So a meta-analysis just refers to taking already published research and looking for connections. So you might find that many published studies all have the same basic conclusion, and based on that similarity, you can connect those dots and develop a theory. The last kind of non-experimental study I want to mention is archival analysis, which will allow you to basically take the data that somebody else has already produced and test a new hypothesis on their data. It sounds a little bit like plagiarism, but it's not really. Yeah, of course, you need to ask permission of the person who created that data in the first place. But assuming they're okay with it, it's perfectly fine to reanalyze data to test a new hypothesis, if you'd like. Now, there are some definite limitations, definite problems when it comes to doing any kind of study. One of which would be researcher bias. Most researchers, you could also call it experimenter bias, most researchers know what kind of outcome they want. Like, they created their hypothesis and they're testing it, but they know what they want. They know what they want to see, and they're hoping that the participants perform in a certain way so that their hypothesis will be validated, right? So researcher bias just refers to this fact, and the fact that researchers will oftentimes unintentionally, alter the participants' data in ways that they are not even aware of, or maybe just treat the participants in certain ways that could lead them to come to that conclusion that the researcher is hoping for. It's just basically whenever the researcher is oftentimes unintentionally altering the results of the study so that their hypothesis will be validated. And this is really unfortunate that this happens, and it is thankfully avoidable by doing certain kinds of methods that I'll talk about in just a moment. Before I do, I want to mention there's a second source of bias. So there's that researcher bias, and then there's also what's called participant bias. So this is when the participant is aware of what the researcher wants and feels pressured to, conform, to produce those results the researcher wants them to do. You could also call this the courtesy bias, and this is something people just naturally have. Like if somebody, imagine that you're participating in some scientific experiment and somebody sits you down in a room and asks you if you see a pink elephant. You're going to say no, there's no pink elephant in the room, right? But what if they ask you again and again and again, and they just keep asking you, do you see a pink elephant now? Do you see a pink elephant now? Eventually, you're going to start thinking, they probably want me to say yes. So eventually, you probably will say yes. It'll be really awkward if you don't say yes, because you're feeling that pressure. You're feeling that kind of social influence to conform to what you think they want you to do. So the ways that you can try to eliminate these sources of bias are by using what's called blinds. There's a single blind and double blind. A single blind just means that the participant is not aware if they're in the experiment group or the control group. So if the participant doesn't know if they should see the pink elephant, they won't know if they should say yes or no. So this will help to eliminate that participant bias. But the researcher bias is harder to eliminate. The way you would do that is by using a double blind. And that just means the participant doesn't know what group they're in, and the researcher doesn't know what group they're in either. You might say, well, how can you possibly analyze the data if you don't know what group the participant's in? Well, the answer is you would get somebody else to tell you what group those people are in after you've already collected all the data. So that way there's less chance that you could be biasing those results.